Hello everyone from around the world. Hope you can hear me. Welcome again to our fifth installment of the London Proteomics Discussion Group webinar series on COVID-19 and the role of proteomics. Today we are joined by Professor Ray Isles and Dr. Martin Darnans to discuss their work in this area. As always, we'll be using our Slack channel for questions and discussion, so please join us there and ask any questions or share any thoughts or discussion about the work that you're hearing about. And remember that the Slack channel actually allows us to prioritize questions that you want to hear the answers to, and you can do that by clicking on the thumbs up icon for any questions that you would like to hear the answers for. And also Slack has the ability to allow the speakers to answer any follow-up questions that there may be after the talks are finished. So stick around for that if you're interested. For those that need attendance certificates for this webinar, the details about that will be given after the final talk, so hang around for that. And also we're going to be running a quick survey just to get some feedback on how you're finding the format so far. And thank you to those who gave us the feedback last time. It was very useful. So I would like to also thank the European Proteomics Association, BSPR and YPIC, and also the London Proteomics Discussion Group Committee for all their help and support in setting up this webinar. We'd also like to thank the London Biological Mass Spectrometry Discussion Group, the London Metabolomics Network, and also the News and Proteomics Research blog for promoting this event. And we're grateful to Imperial College London for providing us with the webinar support. And of course, a huge thank you to our speakers for their time today. We're pleased to also announce that our next webinar in the series will be on Friday, the 12th of June, 2 p.m. British Standard Time, and it will be featuring Professor Marcus Rolzer discussing his work on clinical classifiers of COVID-19 infection identified by ultra-high proteomics. So, first up today, we'll be hearing from Professor Ray Isles. Professor Isles is Dean of Abu Dhabi's University College of Health Sciences. Along with this role, he's the Chief Scientific Officer with MAP Sciences, a company that develops medical diagnostics and digital diagnostic applications, where he's developing IP to market biotechnology tech transfer in cancer, hematology, and prenatal diagnosis. Professor Isles is also a visiting professor of forensic science at King's College London. And with that, I will pass over to you, Professor Isles.
Thanks very much indeed. Hopefully I've got control now. Just click over. There we go. Right, I'm just going to get straight on to the subject in hand. For about 6 years now, we've been trying to, or in fact, successfully moving the use of MALDI-ToF as a research tool into the clinical arena. And all of you who are familiar with mass spectrometry know that there are a number of challenges or variables that have to be overcome. Those familiar with mass spectrometry, and particularly in MALDI-ToF, will know that this includes optimization of the matrix formulation, optimization of the MALDI-ToF MS itself. A lot of settings have to be altered. And one of the interesting things that we found a lot with MALDI-ToF is that it's much better to take the data off the machine and process on other software to get the best out of the spectral data. So when we came to switch our attention to COVID-19 and viruses, we had all of these 5 challenges to overcome. What I'm going to talk about is really the first 2 challenges here, which are chemistry based, all to do with how to process a sample in order to get extremely good spectral data. So I'm going to take you through in sample enrichment and virus envelope disruption, which are the critical steps. These anyone can really fix who've got experience in mass spectrometry. Hopefully it's all going over. So I feel like I'm carrying on from the talk 2 weeks ago, where that one left off with Professor Zintz's group. So, like Professor Zintz and many people around the world, we were looking at acetone precipitation, and we have done this slightly differently. We have moved actually the acetone precipitation, or optimized it for enrichment of the viral particles, and also as a way of getting rid of a lot of small molecules. So we add our acetone at a 1-to-1 ratio with our biological sample at 4 degrees. We then spin it at 16,000 G at 4 degrees for 30 minutes. And this achieves an enriched pellet of your viral particles. You will bring down some large proteins, and of course albumin, our little smiley chap who's always a pain for us mass spectrometrists. But the most important thing is you're leaving behind a lot of small, in fact, you're clearing small to moderate sized proteins from your sample by doing that selective acetone enrichment precipitation. Now, there's another major advantage of using acetone as a precipitant is that it actually deactivates your virus. And there's a lot of published literature that the virus is inactivated by acetone precipitation. So in terms of setting up a clinical assay, this is very important because your biological sample will have active virus, and you may have to manage that within a category 3 containment facility. But as soon as you've acetone precipitated it, you've got dead virus. It inactivates probably by deformation of the viral particle and the proteins, or fragmentation even, where you're breaking it up into smaller micelles. But it is inactivated, and you can actually do the work then on the bench. And if you're if you're worried about it in a class 2, but it's much more safer to actually deal with these viruses. Now, the next step that we optimize is extremely important when we're actually developing this assay technique, and this is the extraction and solubilization of our viral envelope proteins from this cholesterol lipid membrane. We're not interested in the RNA, and I just like to point out to my molecular biology colleagues that you have one RNA molecule, but I could have 10,000 spike proteins per virus. So I'm in a little bit of an advantage over you here, that I've got a lot more material coming out from that one virus. Now, solubilization of these viral embedded or viral associated proteins is quite important. Some proteins, like the nucleocapsid, come off very easily. They're easily solubilized. But others, such as the spike protein stalk or S2 fragment, is embedded within that membrane. And when we've disrupted, we've got a lot of clumps of lipid membrane. So we had to optimize with some very unusual material to solubilize it. I'm sorry, I can't tell you all of that information. It is something that we will let people have, but not quite yet. But we're very good at solubilizing these proteins and releasing them, and they fragment quite nicely so that we get them breaking apart, because most of these are held by hydrophobic, non-covalent associations or dithiothiazole linkages, disulfide linkages, which you can easily break. So our application really was to develop this methodology for looking at the viral proteins in our work with colleagues at Cambridge University at the Laboratory of Zoonotics, so Professor Heaney's group, who were creating pseudoviruses, which are lenti constructs where they have spliced in the spike protein for SARS-CoV-1 and SARS-CoV-2. Having developed our technology on the pseudoviruses, we're able to look in detail at the spike protein. Now, the spike protein has to be proteolytically cleaved, not only in its maturation, just getting rid of the signal peptide, but also at this point here and this point here to become an active, functioning, effective, functioning machinery. So we were able to actually look very much at the fragmentation of this spike protein, and we were able to look and detect straight away S1, so the S1 protein here, which came up beautifully, and the S2. Now, unfortunately, S2 was coming up underneath albumin, and when we grew the cells, the pseudoviruses in serum-free media, we were able to see the S2. But of course, when we add dithiothiazole, we're able to find these other crucial cleavages which were occurring within the spike protein here, which fragmented into S2A and S2B, and we could actually resolve both of those peptides very nicely. So these were being held by disulfide linkages, so that's how we were able to resolve them after our extraction, and including dithiothiazole in our extraction buffer. Now, the important thing about this is not just what information it can give us about the spike protein and its nature in terms of developing antibodies, vaccines, but the whole technology suddenly gave an opportunity to look at viruses in biological samples. This is just some of the key proteins that we could start to look for to identify viruses. And as you can see, all of them have some kind of cleavage in their spike proteins. Most of them, I'm going to sort of just indicate, is that they tend to cleave to expose the fusion peptide. This is a coronavirus here where they only identified one cleavage site. I suspect that, like all the coronavirus, they have to recruit an external protease to cleave here to expose the fusion peptide. So there's a lot of cleavage events occurring. All of these are different, and all will produce a mass spec protein profile that is different. And that's exactly what we were doing. So this is MERS coronavirus. There's the spike protein, there's the STA fragment, and then we're getting a host of other proteins that are being released from the envelope membrane of these, the lipid membrane of the envelope of this virus. There's also a lot more smaller proteins that are being released as well. But just to give you an example of the difference, this is H1N1 influenza or swine flu. You can see we've lost that spike protein, we've lost actually the S2A protein, and we're starting to pick up hemagglutinin and neuraminidase fragment proteins. And again, a lot of information here. Now, as we started to move from pseudovirus onto live virus, we started to get really interested not just in these larger ones, but also in these low to moderate size proteins that are being released. Now, what's fantastic is that the peak intensities are huge, and it's become very characteristic. For example, this lane here, if we look at the not just the spectra, but also the pseudo gel view of the mass spectra, which gives you lanes, it makes it much easier to see things. But here we can see in the sort of 6,000 to 7,000 MZ region a characteristic pattern that is identifying LCMV or Lassa fever virus. Here, these are two H1N1 influenza or viruses that the vet school had, and we were able to clearly see that they have similarity markers. But as I said, it's very lucky that they actually had two variants. They had the 1934 outbreak of influenza A, H1N1 Puerto Rico, and also the 2009 pandemic variant, which was this is an English isolate, which was ravaging the world at the time. But what's really interesting is, look, we can identify that's H1N1, but there are some peptide changes that would indicate that there are differences between these two strains. So what we can do is distinguish between viral families, genus, species. So here we have, again, the H1N1 pattern. This is the LCMV pattern that we tend to get. These are our SARS pseudoviruses. We can see there's a difference here between SARS-2 and SARS-1. I don't know what that is at the moment, but there is clear pattern changes. And it gives you the opportunity not only to look for SARS, but you can look for a whole host of different viruses and match and decide what they are. Now, the application of this findings and this technology is beyond just looking at COVID-19. But at COVID-19 pandemic, we have to look to see what the use potentially now and in the future. At the moment, we're moving towards a back to work scenario. The governments are all talking about track and trace, and you have to screen an awful lot of people to do that effectively. It has taken 4 months for the UK to achieve sufficient testing systems. We're about 100,000 a day. It's actually been quoted that we need to be doing something more near to 28 million samples a day for effective track and trace. Yeah, how we're going to get there. The reliance is on PCR technology. Immunoassay tests are not really available yet and not reliable. Costs are high, and worryingly, we're dependent on other nations for many of the reagents. Now, the plus is that the test is specific for COVID-19 and highly sensitive. But you can't use that same test on a new virus outbreak, and it could give false positives if there's a significant mutation. It's not easily adapted to other clinical needs such as viral load, which you can do using mass spec. It's quite easy to sort of do a quantitative or semi-quantitative look at viral loading. Sorry. Now, I don't think anyone, even the UK, the USA, could afford another pandemic within a short time span. The effect this pandemic is going to have on developing nations in terms of their economies is going to be huge. And whether they're going to recover or not, or how they recover, is really going to be down to testing, tracing, and shutting down infection. Given that increasing global connectivity, I'm afraid we're seeing more and more zoonotic adaptations, and the probability of getting a new viral outbreak is getting more and more likely. We have seen 3 coronavirus zoonotic transitions in the last 17 years. And certainly we were warned about the current pandemic back in 2007. The big question is, could any nation, not thinking about the poor, but the rich countries, could they cope if the virus had the infectivity rate of something like COVID-19, but the lethality of Ebola? COVID-19 is only killing 2% of those infected. I'm sorry, that's very flippant. I shouldn't be. That's a huge number of people. SARS-1 kills 10%, Ebola 30%. Now these are figures where you've got a fully functioning coping health service. If you had the infectivity of COVID-19 and these lethalities, we really would be in big trouble. The question is, could our technological approaches that we're currently using cope with such a future eventuality? Immunoassays have fallen at the first hurdle. PCR has risen to the challenge, but what is the financial costs? And could it cope with more virulent new virus to achieve that current testing results? And I could list and go through all the kinds of concerns that we have on the reliance of just PCR type testing. Now, what is needed for global biosecurity is a test that can readily identify the emergence of novel viral infections, so not be over specific. We need to be able to produce results within an hour, and a test that can be deployed at ports, airports, as well as our hospitals. We need something that can be rapidly applied to mass screening, so that means it cannot be expensive. We have to have technologies that the running costs of which are very low if you're going to be screening millions of people. We can't really be dependent on very expensive overseas reagents. It's got to be very simple reagents to actually do this. And on top of my wish list, you know, we've got to have non-invasive, minimal invasive sampling of the population. So it's a big ask of the diagnostic world to come up with all of this. Sorry. Now, what I've done is given a little bit of comparison of PCR technology, antibody technology with a MALDI-ToF protein mass spectrometry approach, which could be an LCMS system. But the features are that the PCR is highly sensitive detection for virus. Trouble is, development is several millions of dollars, takes 6 to 18 months, maybe quicker. The costs per test per sample could be quite high, and the time, you know, we're still talking about 48 hours on average. I know it's meant to be 24 hours, but my personal experience, it takes 48 hours from sample to result. Antibody tests, well, they're a lot cheaper, they're a lot quicker, but it's going to take a long time to get there, and there's still a lot of money to actually develop the test. And in clinical utility, it's picking up antibodies rather than the antigen. What I'm saying is that a mass spectrometry approach, development cost could be lower, development times could be very quick, low cost, fast running times, and a host of clinical utility in the use of a mass spec based technology, because you're not just looking at a specific, you might be able to pick up a whole host of viruses that might be arising. So after 4 weeks, we progress beyond proof of principle to ready for phase 1 clinical trials. That's how long it's taken us to do this. We're a little bit of a halt while we're waiting for live COVID-19 virus and samples to come in, which we now have. So the proposed home sampling would be a gargle, 10 mils of gargle solution. It could be just water, and it is just water, in fact, spit into a tube. 45-minute preparation time where the acetone is added, precipitation, destruction buffer, and placed onto your mouldy plate. We use the Shimadzu 8020 mass spectrometer, and this one is a very fast benchtop instrument, takes about 3 minutes per sample, and the data analysis can be very rapid. Set up the program just to look for particular markers like the S1 and the S2A as a first pass, and then look for the other markers afterwards. So very rapid diagnostics. I'd like to give acknowledgement for people who are working 24/7 for weeks on end to get this up and running. That's Raminta from my lab, also my eldest son Jason, and in particular Professor Jonathan Healy and Dr. George Carnell from Cambridge University Laboratory of Viral Zoonotics. Funders, I'd love to put up that we were supported by government agencies or big charities, but no, we haven't. This has all been funded internally. So availability of resources from Cambridge, we fitted in. And in Map Sciences, I have to particularly thank my business partners, Ziad Makazumi and Tarek Makazumi, who at the beginning of this crisis believed that we could make a difference and put £250,000 available for us to get another mass spec, get the work done. And I'm really grateful for them. Also for NICEAD for personal support of myself. Just to give you a little bit of hint where we are, this is COVID-19 virus, the Wuhan isolate that started all this. Just 100 microliters of tissue culture media, and it contains fetal cast serum, acetone precipitated, and using our destruction, optimized destruction buffer. And there's the S1 fragment, there's the S2A fragment, a whole host of interesting other large glycoproteins, and then we get a lovely fingerprint down in this region. We're Map Sciences, and you can contact us on info@mapsciences.com, and happy to answer any questions.
Thank you very much, Professor Isles. That was a really interesting talk, and nice to see, you know, that there's potentially some different options out there to PCR in the future for detecting this virus. We've got a few questions on Slack. In the interest of time, I think I'll just pick one that has been liked a few times, so people want to hear the answer. So Benjamin has said, great talk, completely agree that added benefit is the agility of this mass spec assay to deal with viral mutations. That being said, given this top-down method and your ToF data, what degree of mutation can you confidently detect? For example, can you go down to single amino acid changes?
Yes, depending on the amino acid, because the mass spec is that accurate. I actually surprised myself when we started to look at the complete range. We were just looking at large glycoproteins at first, but when we went down to the smaller mass region, from 6 to 15,000 Daltons, you will pick up a single amino acid change if it has a sufficient mass change.
Okay, thanks for that. I think we've got a few good questions here, but I'm very sorry to those people. We're going to have to move on in the interest of time.